Thanks all for coming. We're really excited to hear this evening's lecture by the Kavli Prize winner, Dr. Professor David Julius, and his interviewer, Frank Sesno. I think some of you have heard him before. Before we get started, or as we get started, I wanted to just show two videos from Cynthia Friend, who's the president of the Kavli Foundation, which sponsors part of this, and also from Lisa Ulvers, who's the president of the Norwegian Academy for Science and Letters. So we'll start with those and then we'll come back.
My name is Lisa Ulvers, and I'm the president of the Norwegian Academy of Science and Letters. I'm sorry for not being with you at Carnegie today, but as some of you may know, we are preparing the announcement for the 2024 Kavli Prize Laureates on June 12th. One of the activities related to the announcement take place in Oslo today. It is indeed a great honor for our Academy to cooperate with the Kavli Foundation and Carnegie's Institution for Science for today's event, where the Kavli Prize Laureate David Julius will give his lecture on the molecular mechanisms of pain sensation and heat. Professor Julius received the Kavli Prize in 2020 with Ardem Pataputian for, and I quote, the Kavli Prize Committee in Neuroscience, their transformative discovery of receptors for temperature and pressure. When the Kavli Prize is awarded, we are so privileged to have King Harald V awarding the prize to the laureates. For each of the three Kavli Prizes, astrophysics, nanoscience, and neuroscience, a prize committee is appointed by our Academy. Members of the committees are world-leading scientists within their respective fields. The committee members are chosen by the Royal Society, the French Academy of Sciences, the Max Planck Society, the National Academy of Science in the US, and the Chinese Academy of Sciences. The high reputation of the Kavli Prize is due to the solid work by the prize committees. Professor Julius attributes his success to the mentors he had as a young scientist. Now, leading a prestigious lab at UCSF, he is the mentor of his own group of students. Many of them now run their own successful research groups, thus carrying on the legacy of both David and his own mentors from his formative years. He writes in his autobiography, the Kavli Prize hopefully inspires and reminds us about the importance that intellectual pursuit, discovery, and basic scientific research play in the vibrancy, health, and general well-being of our world. This is at the core of what the Kavli Prize stands for. And as a fellow scientist, I could not agree more. I hope you'll all enjoy today's lecture and look forward to future collaborations.
Good evening, and thank you for being here tonight. I'm Cynthia Friend, President and CEO of the Kavli Foundation. I'm very sorry I can't be with you tonight personally, but I nevertheless wanted to take this opportunity to provide some context and background about the Kavli Foundation. The Kavli Foundation was established in 2000 by Fred Kavli, a Norwegian-born American entrepreneur and businessman. He articulated an incredible vision, and that was to advance science for the benefit of humanity. It doesn't get any better than that, in my opinion. After consulting with university and scientific leaders, Fred identified 4 scientific fields to support: astrophysics, nanoscience, neuroscience, and theoretical physics. Today, we at the Kavli Foundation strive to support research in these fields that has potential for transformative impact. We also strive to improve and enhance the connection between science and society, a consideration that's more important now than ever before. The Kavli Prize, in fact, was created to this end, and it honors scientists and their scientific achievements to increase their visibility with society. The Kavli Prize is given every 2 years and was starting in 2008, and it is a partnership among the Norwegian Academy of Science and Letters, the Norwegian Ministry of Education and Research, and the Kavli Foundation. Tonight we welcome Dr. David Julius from the University of California, San Francisco, who was awarded the Kavli Prize in Neuroscience in 2020, along with Professor Artem Patapoutian, who's from Scripps. They were also both honored as Nobel laureates subsequently. Dr. Julius' research on the sensation of pain and heat will be featured tonight, and it's a topic I'm sure all of us can relate to. I expect that you'll have a very stimulating conversation. Before I hand off the program to Dr. Julius to discuss his science, I did want to take this opportunity to thank and acknowledge our partners, in particular Frank Sesno, who's our moderator this evening, and also the Carnegie Institution for Science, who's been our partner since 2009 to feature selected Kavli laureates. The Carnegie Institution for Science has created a very engaged audience for science through the Capital Science Evenings, like the discussion tonight, and we thank them for that. I understand that my colleague, Dr. Eric Isaacs, the president of the Carnegie Institution for Science, will introduce Dr. Julius. So I will hand you over to him now, but let me close in thanking you and wishing you a wonderful evening.
Thank you to Lisa and to Cynthia. So we're really excited and honored tonight to be able to co-host, partner with these organizations, Kavli and the Norwegian Academy, but also with the Norwegian Embassy here in DC to present to you another one of our Kavli lecture series. They're a very great series, and in particular, we're delighted that Frank has agreed once again to moderate. And I am really delighted tonight to introduce Dr. David Julius, who really remarkable science that he's done, and you're going to get to hear about a lot of it tonight. He is the Morris Hertzstein Chair in Molecular Biology and Medicine and Professor and Chair of Physiology at the University of California in San Francisco. His research is really interesting. You've already heard he's interested in pain, sensory perception. Sensory perception depends on the mechanics, the biological, mechanical, electromagnetic parts of the body. So the great discovery that Dr. Julius or David made was to actually identify these particular type of proteins called their channels, their conducting channels that will enable charge to start the pathway from the skin, where you may touch something hot, where you may be exposed to something that's particularly abrasive, and travel up to the brain, perceive it, and then come back. So there's actually a very deep, interesting whole system within the body that does this. And in particular, it's this one protein, which does the discovery, if you will, of that initial pain. One of the very interesting things about what David does is he doesn't use he uses the most interesting materials such as capsaicin, which is, you have a pepper, it burns your mouth, you touch it in your fingers, it burns, you feel that. It's a stimulant for these particular types of proteins in the skin, basically at the end of the neural system. And so he's been doing that. He's also using what I guess menthol from mint, which has a different kind of sensation. So everything you feel, whether it's pain, whether it's temperature, your body reacts, is David's interest. And he's been studying this for years, and it's what brought him to the Nobel Prize. But more importantly than the Nobel Prize, he first won the Kavli Prize in 2020. The Nobel Prize was sort of a second prize to the Kavli Prize. And so, so we're very excited by that. David earned his bachelor's degree at MIT, and he got a PhD at Berkeley. He went to do a postdoc at Columbia and worked at the Howard Hughes Medical Institute professor. And finally, he ended up back at UCSF for a couple of years there, and that's where he did a lot of his great discovery work. He's received lots of honors. I've already mentioned the Kavli and the Nobel Prize, but many others, which are too extensive to mention here, so I won't. The conversation tonight is between Dr. Julius and Frank Sesno. Some of you may know him here in the city. He works at GW. He's also got a deep history in media and in journalism. He's currently the the director of strategic initiatives at George Washington University and the creator of Planet Forward, which is a user-driven platform for television, for computers, essentially to look at how highlights and innovations for sustainability. And he won't be able to talk about that tonight, but that's a whole nother interesting discussion. And prior to this, Frank was spent 11 years and 11 years at the Media and Public Affairs. He was head of Media and Public Affairs at GW, and he spent 20 years at CNN, where he served as the White House correspondent and amongst other things. And so you can talk to Frank about his wonderful interviews after this talk. But right now, what I want to do is just invite Frank and David up for a discussion. I think you'll find it fascinating.
Well, good evening, everybody, and welcome, and good evening, David. Thank you for being here.
Good evening to you.
Pain. We're going to talk about pain. Show of hands, how many in the audience have experienced pain? Okay, so there's a little connection here.
Good, yeah.
You've, as Eric was pointing out, you've identified sort of molecular machines and pathways that help us understand how we feel heat, cold, inflammation, and related physical sensations, AKA pain.
Yeah.
In many cases, and there are different types. And we'll get to some slides that are going to help you explain that. But start by, like, what is pain? When you say that, and when we think about that, is it a thing? What is it?
Yeah, so pain is a sensory modality. You know, it's part of a sensory modality that we call somatosensation, which colloquially we refer to as our sense of touch. So the sense of touch includes things like being able to, you know, feel a light touch, so you know what textures are like, the ability to sense changes in temperature, changes in pressure, and then, and the ability to detect certain chemicals in our environment or in our bodies that can be, in particular, injurious. So pain is really like the extreme arm of touch sensation, if you want to put it that way. So it's a component of touch, and the reason that we have it, I think it's something that we always think of as a negative function, but it's really to tell us when we have or when we're about to experience injury. It's a protective function, and I often say when I lecture about this, of course I'm biased, but that among our sensory modalities, you know, vision, hearing, etc., that pain sensation, it may be the most important for our survival and well-being.
Well, hence the title of the talk here, of our conversation, the purpose of pain.
Right.
So the purpose of pain is
to protect us.
to protect us.
And, you know, you can see that very clearly when you think about or consider people in whom the pain system's not working well. So there's a couple of different examples. One is a very rare example where people have these rare congenital, mutations that, in this case, are in a gene that inactivates a gene that encodes one of these ion channels, not one we've worked on, but another one called the sodium channel, that's required for sending electrical signals along neurons that are involved in pain sensation. And those people have what's called congenital pain insensitivity. There are different descriptions of it, but that's generally what it means. And children who have that don't understand pain. You know, they don't know how to protect themselves. So they may be, for example, the child in school, be very rare, who, you know, is playing with a pencil and puts the pencil through their hand, or goes in the sandbox and gets sand in their eyes and doesn't know it because their sensory nerve fibers in their corneas are not working, and they get corneal abrasions. And so you have to teach these children, you know, what's dangerous and what's not, and it can be very challenging. And then the more common thing is people who have diseases like diabetes, where, if, you know, you may have sort of an accentuation of the pain pathway, but then with time it diminishes. And, you know, that's a common thing where people with diabetes have to check their feet for, ulcerations and cuts because they may not feel them. And this could lead to infection and other kinds of things. So those are examples of how important it is to have a functioning immune system.
Before I put up this first slide, I want to come back to what you were just saying about pain. The purpose of pain is to protect us.
Yes.
I get that. We get that, right? You don't touch the hot tea kettle because you burn your hand. You don't put the sand in your eye. It hurts because it hurts your eye. But there is pain, some of it's chronic pain, some of it's other pain, it's not protecting us, it's
Right,
just a
exactly.
pain,
Yeah,
right? I mean,
yeah.
we're living
So,
with it.
right.
So
So this is the, what I call the Janus face of pain. You know, you need it to protect yourself, but then pain becomes, you know, what people in the field say is maladaptive, which basically means you have pain when it's no longer useful as a warning system. And, you know, we all experience that, you know, sciatica, arthritic knee pain, you know, you name it, lower back pain, migraine, itch is kind of a component of that as well. So yeah, the plus and minus of pain is that it's a really important protective system, but when it goes awry, you can be left with pain that's debilitating and no longer useful as a warning
we'll
system.
talk about that and
Yeah.
incorporate your questions, but let's go to this first slide as we talk about pain, where it comes from, how you
Okay, yeah,
follow it, map
Uh-huh.
it,
Right.
what's happening in our systems, as Eric was discussing.
yeah.
Tell
So
us about
pain,
this.
you know, there's basically like two, I don't want to get too technical, but two aspects of pain. One is the initial act of sensory nerve fibers. So imagine sensory nerve endings in your hand or in your finger that have to detect a stimulus. That's how all sensory systems work. First, there's a stage of stimulus detection. So in your eye, for example, that's something we call phototransduction, where photons activate molecules, receptors in your eye, and then that gets transmitted to your brain. So you can interpret that stimulus as a visual image or light as a visual image. And in the pain pathway, there's a similar thing. You have to detect stimuli, which can be, for example, a change in temperature, change in pressure, like you whack your finger with a hammer. And this is also true for the other, you know, for the other aspects of touch that are non-painful, like light touch, et cetera. They all begin with these receptors, these molecules on the surface of your nerve terminal that detect certain stimuli. So like I said, temperature, heat, cold, pressure, and chemicals. So there are a lot of things in our environment that we have to be aware of and avoid, like you know, noxious chemicals. And there are receptors for all these things on these nerve endings that innervate your eyes. You're in the touch pathway, in this pain pathway, almost, you know, your whole somatotopic surface, meaning your skin, everything's innervated.
When you say receptor, just
Yeah.
explain that.
Okay.
What do you mean?
Yeah, and so a receptor it's a protein it's a molecule, a protein that will sit on the surface of these nerve endings, and there are different types, but the ones that we work on are called ion channels, which Eric mentioned. And what these molecules are like is essentially, and actually if you look at their structure, which we'll look at later, they really look like this. So they sit on the surface, so every cell has a membrane around it that, you know, delimits it from the outside world. And in the nervous system, the currency of communication is electricity. So what has to happen is these stimuli, like temperature or pressure, have to be converted into an electrical signal. So that signal then gets sent along the nerve fiber, that in this case goes to your spinal cord, and then sends that message to the next neuron in the spinal cord, and then through a whole way station of neurons to your brain, so you can perceive the stimulus detection out here as something cognitively that you
And you have in the top of this chart, brain pain networks.
Right, exactly. And so these molecules that serve as receptors, in this case, what they do is when they're activated by something like heat or cold, they generate an electrical signal. And the way that they do that is they sit on the surface of these cells and they sort of form essentially little donuts in the membrane. And in the resting state, meaning when they're not activated, the donut hole is closed. When they're activated by something like pressure or temperature, the donut hole opens and it allows ions to flow across the membrane. Ions, things that, you know, are essential for our life, like sodium, potassium ions. And this generates a regenerative electrical current through something called an action potential that then gets sent to the spinal cord and to the central nervous system. So this initial phase of stimulus detection is something we call nociception. And then nociception is turned into pain when those messages get sent to the brain, and you can interpret that as, you know, emotionally, cognitively, et cetera, as what that stimulus means.
Where are we feeling the pain?
That's a bit of a philosophical question, but we're really feeling the pain in the brain. Yeah.
Right. So,
but they're important. But of course, the detection is the first essential step. And there's two things I want to mention. One is that, as drawn here, everything that's being detected is in the outside world, right? Change it. You know, you touch a hot stove, you touch a cold ice cube, you hit yourself with a hammer. But we have sensory nerve endings, not just in our skin and our eyes. We have sensory nerve endings also in the organs in our body. And we all know this because When you drink too much water and you're sitting here on the stage, you say, gee, I really got to go deal with that. That's because you have sensory nerve endings in your bladder that are measuring the stretch. You have sensory nerve endings in your gut, something we're very interested in now, we can talk about later, that are involved in sensing, when there's dysbiosis or other things happening in the gut that you need to know about, and detecting chemicals that your own body or microbes produce that are warning signals.
So let's take a look at this.
Yeah.
This shows us some of these very things you're talking about. There's that first slide there, somebody reaching to the back. I'm familiar with that. That's why I stretch every morning.
Right.
But I see somebody with dental pain.
Yeah.
Is all pain the same?
Probably not, almost certainly not.
Do we know?
And we're starting to learn. And I think we know that from our own experience. Well, one, it's not the same cognitively, because part of your brain is wired to know not just what type of pain it is, heat, cold, pressure, and sometimes it's hard to, those things get conflated. Sometimes it's hard to know, but we also know where in the body that pain's being experienced, because there's a region, an area called the somatosensory cortex that maps where those fibers come from and tells you like the pain is in your mouth or in your knee. But sometimes it's confusing and it's hard to know where it comes from. But, some drugs are good at treating certain types of pain, but not others. And that tells us that there are probably some differences in pain mechanisms. And one of the big goals, I think, in the pain field now is to understand what makes different types of pain different. So I often say that we use this one term pain, like we use cancer. I mean, I don't think anybody today would say that all forms of cancer are the same. There are some common elements, certain oncogenes and other things that are promoters of cancer in different tissues, but glioblastoma is different than cancer of the lung, et cetera, et cetera. And migraine pain and GI pain, gastrointestinal pain or lower back pain, almost certainly share some commonalities, but have differences as well. And understanding what those differences are and what molecules and cell types are involved, I think is critical to thinking about developing therapies that target different types of pain.
So therapies, which is where we go next. I mean, one of the most fascinating, important parts of your research is whether and how it will help us figure out how to treat pain in different ways, with potentially non-addictive medications or other more natural forms. So let's just go to that for a minute, and that is the question of how do we manage pain? And how do we do it now?
Okay. I think we animated a slide, so go to
Is that the wrong one?
No, it's just I had animated
not
this, so I forgot to
advanced.
take it off. Okay, so how do we manage pain now? Well, we know how we manage pain now. Mostly it's through, a very small number of chemical classes. There's the non-steroidal anti-inflammatories, aspirin, ibuprofen. Acetaminophen is an antipyretic. And then there's opiates. And those are sort of the two main classes. There are other things now like gabapentin and some of these gabapentinoids that are sort of, somewhat successful at treating certain types of pain. But, that's kind of most of what we use. And certainly these things, that we use every day. So, some of them are wonderful. I mean, I took some, Aleve or aspirin or something a while ago for some soreness, and it was great. But
it worked.
Yeah, it worked. And opiates, can be amazing at treating pain. But as we know, they all have their different side effects. Things like, well, for these, liver and kidney toxicity. So you can only use them to a certain amount. Of course, opiates have all the, things that we know about: tolerance, addiction, respiratory depression, constipation, et cetera. So they work, but they have some really limiting side effects.
And there are, as you say, there are side effects to these things. So it's a trade-off between what works and what doesn't. I don't know how many times I have a conversation with people who are wondering, how often should I take acetaminophen or ibuprofen? What is that? So is your objective in your research, we'll come to it in a minute, but is your objective to get rid of these things so we don't need these anymore?
So what I should say is, and to some degree this is holding my banner here, but I'm a basic scientist. And I think it's important to say my objective is to understand how things work. And it's curiosity-based science. I know that sounds a little selfish, but
that's what it is.
My objective is to satisfy my curiosity and understand how things work. And it's not really a selfish goal because until you understand how things work, you can't really solve it.
But you know that if we can understand how
Yes,
these things work.
but in the end, part of the reason that I've chosen to work on this as opposed to something else is that I know that if we can figure out how things work and identify new players in this pathway, that's really the only kind of, rational mechanistic way forward to developing new treatments for pain
So
sensation.
what your research does suggest, and where it's headed, is that there are avenues to develop
Yeah.
safe,
Right.
non-addictive.
Well, we hope so. Yeah.
All right,
Right.
and some of them natural, like,
Yeah.
let's hear it for chilies.
Yeah, yeah. So actually, I should say, so if you've noticed on the initial slide where we talk about the pain pathway, what we chose to do for a variety of reasons, but for one that I'll describe in a minute, is to look at molecules, in these case receptors, that are present at the very beginning of the pain pathway. Okay, so you could so pain, as I told you, is this, circuit map that involves neurons in the periphery as well as neurons in the spinal cord and in the brain. And people study this at all levels, neurophysiologists and behaviorists. They're studying all aspects of the pain circuit. But we chose to study the very beginning aspects of the pain circuit for a number of technical reasons as well. But, part of that motivation, part of what motivates that is the idea that if you can find molecules that are found early in the pathway and very specifically in that pathway, then you have a better chance of developing drugs that can act to suppress pain without side effects. So the problem with opiate drugs is that there are opiate receptors all over the nervous system, right? So there's opiate receptors, along the early parts of the pain pathway, but there are opiate receptors all over the central nervous system. And in fact, there are opiate receptors in a brainstem region that controls breathing. So that's why when people overdose on opiates, they stop breathing, and that's what kills them. And so the idea, and the same thing with acetaminophen and NSAIDs, is that they target molecules that are present, not just in the neurons in the spinal cord, but also in these other internal organs, liver and kidney. And so the idea was if we can find molecules that are more specific for the very beginning of the pain pathway, that might give us an edge in trying to identify drugs that would have much more specific effects.
What then might some of the alternatives to these be based on those pathways? And what we're
so like in our case, for example, so we've looked at these, we focused on a number of molecules, but we focused on 3 and other labs have focused on others that are expressed in these neurons that carry out this early process of nociception, detecting a noxious stimulus or transmitting that information to the spinal cord. And so the alternatives would be to develop drugs that block the action of those molecules to really sort of prevent the initiation of pain sensation. And, so far it's still, there haven't been any successes so that you can walk into, CVS or Walgreens and buy a drug. But, it's still pretty early in this field. And I think there's some progress being made. But, drug development and drug design is a pretty tough road. And but in any case, that's the logic, is to find new molecules.
Go into that a little bit more. If that drug were to be developed,
Yes.
what would it be doing?
Oh, what would it be doing?
To these pathways and to these proteins,
right,
to relieve us of pain?
Okay, so in some of the molecules we work on, they're involved not just in detecting the initial pain, but they're also involved in generating inflammatory pain. So when these neurons are activated, let's say at the periphery, and you activate them, they send a message to the brain that tells you you've just done something that's, you've interacted with something that could be injurious or painful. And then when activated, those neurons also release substances that initiate this process of inflammation. And then that drives pain hypersensitivity. So an example of that would be, inflammatory knee pain is acute pain, but it's also an interaction. It's also where inflammation is happening and sensitizing the whole nervous system, the whole pain-sensing nervous system. And we know this because if you eat a hot chili pepper, which, as you heard, activates some of these receptors that we've been working on, you feel something that's very painful, and then your lips become very swollen and pouty, and that's because it's initiated an inflammatory process already. And that inflammatory...
Some people can do that and some people can't, right?
Yeah.
Like different people react in different ways.
Yeah, yeah, right. And that inflammation that occurs then acts back on these nerve fibers to sort of turn this cycle of increasing pain. which then increases inflammation. So the idea would be that if you can inhibit those receptors that we've talked about, you can stop that process of what we call neurogenic inflammation and prevent the kind of wind-up that leads to an enhanced pain
It's
response.
interesting. So, and different people have different, how many, anybody in the room just love super hot peppers and you can actually eat them? A couple of, so what's going on with them that other people,
Maybe there's some mutants. We should get some of your DNA. You know, some of that is, I mean, there's a whole aspect of pain that we haven't spoken about and that I'm not an expert in, but that is the psychological aspect of pain. It's a very, you know, integrative and cognitive experience. And I think some of us just have, you know, different thresholds for pain. And of course, you know, when you eat a chili pepper and once you've gained experience, you know what to expect. So it's not something that, it's not like hitting your hand with a hammer, you know, it's something that you can deal with to some degree. Of course, we all kind of run out of road physiologically, but, and, you know, you're anticipating this. And, you know, in some ways, as we know, there's a fine line between pleasure and pain. And some people find that pleasurable. But for those of you guys who really like hot chili peppers, you would not get the same joyous response if you forgot to wash your fingers and stuck your finger in your eye and put some, you know, that would be pure pain experience and you would not enjoy that.
all right,
So
so
it
let's,
depends on context. Pain depends on context. It depends on expectations.
well, it's interesting, and we're
and
going to
you-
get to the,
you know,
but some people, and I know this, a friend of mine, he's a doctor.
Yeah.
And he works a lot with people with lower back pain.
Mhm.
And he talks to them about working through your pain.
Yeah.
And some people can do that and some people cannot.
Right.
Is that purely psychological? Is some of that actually physiological? I mean,
Yeah,
what do,
probably both, I think. But I think that there is a psychological component to it.
and what is your
And people have different
molecular
thresholds
machinery
for
tell us
pain.
about that?
Nothing yet. Yeah. I mean, that's a much more, you know, people who study that are really studying, you know, they're really at the juncture of neuroscience and psychology, and also using imaging methods to understand what's happening in the brain in terms of those signals. But I think understanding the cognitive aspects of pain is probably the most difficult component of this.
I'm seeing tarantulas and snakes here.
Yeah.
And we're supposed to talk about natural products and medicine. So I suppose if you turn the tarantula on me, I will forget about the hammer that I just hit myself
Right,
with.
right,
But
yeah.
what is the connection?
What's the point of this? So, you know, our approach to this, the way that we approached this whole problem in the beginning was to exploit natural products. So we asked, decades ago, for example, people in Hungary found that, because there's a big paprika industry, that capsaicin, which is the pungent ingredient in chili peppers, is a very selective activator of these neurons that are involved in pain sensation. And so that became kind of a holy grail to understand how this works. And likewise, we know that, you know, menthol is a cooling agent, and so it likely has something to do with activating neural pathways in your body that are involved in temperature sensation, cold sensation. And so we started this work by just basically doing chemistry and using these natural products, capsaicin and menthol, to ask, how do they work? Are there specific targets for these things in the body? And if so, once we identify those molecules and the genes that encode them, could we use those as kind of toeholds to understand this whole circuit of pain, and eventually use that as a way to ask if new analgesics could target those pathways, et cetera. So it gave us, you know, molecular and genetic handles to do that. But, you know, we're inspired by a lot of work that came before us. We're not the only ones to use natural products to study the body and physiology, and there's a great history in all of this. And many people, I think, don't realize that a lot of the drugs we take today are inspired by natural products. I mean,
Well,
if you look at
indigenous cultures who have been
Yeah.
using these things and some
Right.
of these things in many, many ways. So have we got breakthrough material here?
Oh, yeah. Well, you know, if you look at the pain field, for example, even long before the things that we've talked about. So non-steroidal aspirin comes from the willow bark. That's a natural analgesic. Opiates, of course, come from opium poppies, morphine. So that's another one, you know, been used
It is
for
a natural
centuries.
product.
And those have also, you know, been used as tools to understand how they work. And so aspirin led to the identification of these enzymes called cyclooxygenases, which are key molecules involved in inflammation. That's why aspirin's good for inflammatory pain. Opiates, of course, led to the identification of opiate receptors, and
Mhm.
understanding how that whole system, how endorphins and enkephalins work in our body. And then, some of the antihypertensives that we take are inspired by molecules found in snake venom. I put this Gila monster on here because work in the Gila monster venom was found has an interesting version of something that we've heard a lot about in the news, this peptide called GLP-1, Ozempic, right? And studies of this peptide, this GLP-1-like peptide in Gila monsters is what gave chemists clues on how to make that molecule more
Do
stable.
you have
so it could
Gila
be-
monsters in your lab? or
No, I don't, but we've had spiders and snakes. And spiders, so, why does a chili pepper make capsaicin? Presumably it's to defend itself against predation by a squirrel or a deer, because, they have sensory nerve fibers, and unlike us, they haven't sort of learned how to appreciate the culinary wonders of the pepper, so they eat it and they don't like it. And, spiders do the same thing. They don't have capsaicin, but they have these toxins, toxins in their venom that bind to the same molecules, receptors that bind capsaicin, and really activate them strongly. So when you're bitten by one of these spiders and you're in a lot of pain, in part it's because they've activated your chili pepper receptors. So there's a lot, and then I put a Petri plate here because, of course, we all know that many, fungal agents have given rise to antibiotics,
Right.
immunosuppressants that we use, like FK506 and all, when people get organ transplants. So, the natural world has been mined for, amazing chemicals that have either become drugs or given us clues. The targets have led to the identification of the targets that have told us what targets to develop drugs to.
Let's take a look at this. Next one here. What's this?
All right, so in my lab, as I said, we've identified these receptors for things like chili pepper. So the chili pepper receptor, called the capsaicin receptor, is, maybe not surprisingly now because of your psychophysics, that it's a molecule that's involved in our ability to detect heat, which makes sense, right? You eat a chili pepper, it's sort of, you conflate that with a heat sensation. And over here is, and then there's another one that's related to this that I don't show you, that's a receptor from menthol. And that's a receptor that allows us to detect coolness and cold. This one called the Wasabi receptor, and they're all related. They're sort of related genes, but distinct.
It's called the Wasabi receptor.
Well, we call it the Wasabi
You
receptor.
call it the
It's called TRPA1. They all have these gene names, okay, but we call it the Wasabi receptor. And this receptor is very important because it's activated by chemical irritants. So when there are things in the environment that you have to be aware of, like when there's a forest fire and there's a lot of particulates in the air and then gases that get produced, some of those molecules activate this receptor. So when, when you get smoke in your eyes, someone smoking a cigarette, it's a compound in the cigarette called acrolein that activates this receptor and tells you you should get away from whatever that is.
Is that also all smoke or just a cigarette? I mean, you're just citing that as an example because it's in tobacco or
any burning vegetation, also, vehicle exhaust, a lot of these volatile irritants. And it's also, so you can relate this not only from wasabi, but there's a compound in onions when you chop it called a lysine. And when you're rubbing your eyes in pain, that's because the lysine has volatilized in the air and then gotten into the conjunctive in your eyes, and it activates this beautiful little receptor, and that initiates a pain response.
Are you like the draw at every cocktail party because everybody wants to come talk?
Yeah, they want to know how these things work.
I mean, it's amazing.
So what these things are.
I mean, this is just incredible.
So what these pictures are so one of the goals in the lab is, and related to therapeutics ultimately, is to understand what these molecules look like in 3 dimensions and in atomic detail. And so we've used these new methods of microscopy to see what these molecules look like in 3 dimensions. And this is kind of a cartoon of what they look like. And the reason to do that is to sort of look at all the little nooks and crannies in the molecule and understand either where drugs that we know affect these molecules can bind or where new drugs could be designed to modulate their function. And so in this one over here on your left, you can see this is the capsaicin receptor, and the little red blobs sitting in there are a molecule of, from chili pepper, capsaicin bound into the channel, into the molecule, so we know where it works. And little purple blobs on top are a toxin from a spider that also binds to this channel and activates it. So we get a real molecular view of how these things come together, and that helps people think about how they can design drugs to modulate the activity of the channel. And the same thing's true for this receptor. So that's kind of a very, molecular aspect of what we do.
Could you talk to us a bit about what, some of the big questions are that you're trying to answer now?
Yeah.
What are you in pursuit of?
So one, so there's sort of two parts of my lab. One are the people who really like to play with molecules, and they're in pursuit of understanding how these things work as molecular machines. So just briefly, what I'll say is, so this capsaicin receptor is interesting because not only does it detect spices and heat, but as I sort of alluded to a moment ago, it also has places where it detects inflammatory agents. So when you have tissue injury, let's say in an arthritic knee, and you've generated, your tissues generated these inflammatory agents, they will bind to this receptor and other proteins, not just this one. And that's part of the way that they enhance pain sensation. And so one of the goals is to understand how components of what we call the inflammatory soup, the inflammatory tissue, fluid in your body, modulates these ion channels to enhance their activity. And the idea is that if you can inhibit that, if you can counteract that, then you can think about new ways to develop analgesics. So that's one goal, is to really understand the kind of nooks and crannies of these molecules for drug design. And just understand how they work, because they don't stand alone. They're related to a lot of other molecules in our body that look like this. So the relevance of our work in pain is also extended to other kinds of, cardiovascular disease and things like that. And then the other part of the lab is more integrative, and it's really to understand, getting back to that previous slide, what makes different pain different or similar. And, one of the things that we're really interested in now is understanding not just pain sort of from the environment, but pain that we sense internally. This sort of field that everybody's all, crazy about now, called interoception, which means sensing stimuli from within. And
What is that? Is that pain?
yeah, it can be pain. It doesn't have to be pain. There's a lot of sensations. There's a lot of detection of things that are going on in our body that regulate what we call homeostasis. So for example, when you eat a meal, you have to know, what's happening, you have to detect nutrients, sugar balance, et cetera, and then that information gets fed to the brain to regulate your feeding behavior, satiety, all this kind of stuff. And so there's a big focus in neuroscience now is to understand the crosstalk between the brain and the body. So we always think of the brain as something that, outputs emotion or controls our body in many ways, but there's a lot of crosstalk up and back between our internal organs that regulate, immunity, digestion, feeding behaviors, all kinds of internal processes. And one thing that we're really interested in, but we're focused mostly on how this relates to pain. And one of the models, if you want to call it, or one of the paradigms we're looking at now is to understand what we call visceral pain. So mostly that would be gut pain. And that's sort of, so an example of that would be irritable bowel syndrome. So why are we interested in that? Well, one, there's a lot of interesting biology in terms of how, so there are sensory nerve fibers, as I said, all over your body, and there are sensory nerve fibers that go deep into your intestine and innervate all the little villi in your intestinal tract. And there's a lot of interesting biology in how those nerve fibers interact with cells in your gut. So just from sort of a purely scientific point of view, it's very interesting. But also, it's relevant to disorders that so many people experience.
Such as?
Like IBS, and not just in the gut, but also in, hyperreactive bladder syndrome. And one of the other interesting things about this is that, pain, there are also interesting aspects about sex differences in pain.
You've noted as the disparity.
Yeah, and in GI pain, there's a big disparity. So women suffer from GI-related pain about 3 to 5 times more commonly than men.
3 to 5 times.
Yeah, and it's usually women, in their reproductive ages. So this is like my wife's a scientist, does her own thing in sort of endocrinology, but this is one area that we work on together because she's really focused on sex differences as it relates to women's health and the role of estrogen and other, reproductive hormones in this. And so we've come together to study this and understand what are the basic mechanisms in men or women, but then what is it about women's physiology that makes them more prone to visceral pain. And, the visceral pain is, there's an aspect of it that's acute where, you just feel pain and malaise or, discomfort or bloating. But then there's a whole effect, is true for a lot of pain on your long-term behavior. It generates a lot of anxiety. because there's, social aspects of it and then.
Oh, it's so rapid, right?
Yeah, and so, we want to understand mostly what's happening here and how all that's generated and, what's going on in terms of.
Have you made
molecules.
progress on that?
Yeah, we have actually.
What?
Well, we've identified cells. So in the gut, there are a lot of cells in the lining of the gut that separate what we call, the lumen from the outside. So the lumen's where all the food stuff go through, where the bacteria live. And then, all those nutrients have to go through and they go into this area called, the mucosal region, which is then has a lot of neurons in it, musculature that regulate the contractility of the gut. So in any case, there's a whole bunch of cells that line that barrier. And most of them are, involved in transport of water and other nutrients. But every once in a while, there's a rare cell that's kind of another type of sensory cell that detects different stimuli and releases a lot of transmitters like serotonin and, peptide hormones. So this is where like things like GLP-1 normally in our body come from. And so we've really begun to characterize the interactions between some of these very rare but interesting specialized cells in the intestine and the pain sensing neurofibers with which they interact. And we're beginning to understand what activates them, what activates this pathway. And I think we're beginning to understand what are some of the differences and this is all being done in mice, but male and females that can account for the differences in sensitivity within the gut to pain.
This is amazing, and I'm going to go to some of your questions in just a couple of minutes here. And if I can advance this here so that I can put the QR code back up. If you want to send us a question or weigh in on a question, it's up there already. But before I do that, let's talk about you for a couple of minutes. Okay. You know pain really, really well. You understand this. Like when you hurt yourself, do you just think differently? It's like,
Yeah, probably not. It's like you whack yourself with a hammer. The other day I did have some, I think I needed root canal, but I had some tooth pain. And, I'm just like I think about it a little bit differently, of course. But, the bottom line is that when you have pain, it really takes over your brain. Right. And you're not thinking about anything else except how can I get rid of it?
When you know that and you know what's going on in there, can you like talk yourself down from the ledge somehow?
A little bit, yeah. I can a little bit, but still.
And do you feel the pain?
Depends on the severity.
You feel the pain less?
I'll say I need to find a dentist, Do I feel a little bit less? No, probably not. Probably not.
Interesting.
But it is a reminder of how overbearing pain can be when it doesn't go away.
How did you get into, when you described yourself early on as a reluctant student.
Yeah.
You're obviously not now.
Not now.
But did you wake up one morning and say, I hate pain or I love pain and I want to spend the rest of my life studying it?
No, I didn't start. Like a lot of scientists, you come into things by different directions. But so my background in science is really what I'd call very reductionist, meaning that I've always been interested in how molecules work. There are people who come to neuroscience from a much more integrative point of view, which means that, for example, they may come from the viewpoint of psychology or behavior. And so they're studying, or, studying neural networks, and they're really trying to understand, neuroscience at a more integrative, higher level, if you want to put it that way. I've always been fascinated by molecules. I'm trained as a biochemist and an enzymologist. And so, when I was an undergraduate, I asked how enzymes work to catalyze reactions. When I was a graduate student, I actually worked on yeast as a model organism, a genetic organism, to understand how yeast make peptide hormones, much like we do, like insulin, things like that, and to use genetics to understand the molecules that allow those things to happen. And then, through that work, I started getting interested in asking, well, how do things like peptide hormones work? How do they signal? What do these receptors look like? And I got interested in neuroscience, because of that, but also, I was a graduate student at Berkeley in like the 70s. And so, you couldn't help but think about, how do things like hallucinogens work and, drugs that affect the nervous system and, all these people who were like, using psilocybin or, it was not just Berkeley, being funny about that, but I got interested in understanding how these small molecules could affect behavior and the brain so profoundly. And then also, that's when I really got interested in this whole idea, which wasn't, I was just reading the literature and taking my lessons from other people, but this beautiful thing where people exploit natural products to understand physiology. I mean, when you think about that, it's this beautiful convergence of anthropology, chemistry, and physiology. So over thousands of years, people have to experiment, sometimes probably kills them, but they have to find a plant that does something. Actually, there's a really interesting article in the New York Times the other day about, I think it was an orangutan in Sumatra, who it's like the first evidence that they're actually foraging for natural products and using them to, in this case, treat a wound under the eye, which suggests that, maybe it's a little anecdotal, but maybe in those societies too, there's a passage of information through the generations about the use of plants for medicinal purposes. But in any case, I thought that's fascinating. How do people discover these plants? And then how do chemists understand what it is in the plant, or, venom or toxin that's useful? And then biologists have to come along and figure out, how those chemicals work. What are their targets in the human body? And then you can go full circle and ask, use chemistry now to make drugs, and can we use those again for behavior? So I sort of like that.
I mean,
circle of
well, and you are actually now describing the trajectory of curiosity-driven science.
Exactly.
Right? This
yeah.
is your
and so this
your
is kind
journey.
Of, yeah, and so I got interested in what we call neuropharmacology. How do drugs work? How do they modify behavior? And so, and on top of that, I was really interested in sensory systems. Why? Because all your sensory systems, vision, hearing, taste, describe how you experience your world. And we think that everybody or everything experience, well, we don't really think this, but it's the assumption, that we often don't think about enough, that all organisms or all people, see the world the same way. But of course, we don't see the world the same way a honeybee does, or a snake or whatever. We have different sensory systems. And so how we see the world, the colors that we see, the sounds we hear, is a complete construct of the molecules that we have that detect certain types of stimuli. Dogs can hear in a different range of sound. Old world and new world monkeys see light with somewhat different hues, depending upon what they have to adapt to find like an apple in a tree. And honeybees can see in the ultraviolet, range of light spectrum. And so our world to us and then all that information gets fed in, and what your brain does is it makes an internal representation of the outside world, right? And so we navigate the world based on what our sensory, apparati and all the biophysics and biochemistry tell us we can detect. And we can't see the things we can't observe or experience the things we don't detect. We can only imagine what it's like. A shark can detect weak electric fields in the ocean. We can't really detect that. So we can only anthropomorphize what that's like. We can't really experience it, right? And so I think that's a fascinating part of sensory.
We have some tremendous questions from the audience, so I want to turn to those now. And you get to vote on one another's questions, so I can see the most popular questions, and might start with a few of those.
All right.
So here's one right at the top. How neurologically sophisticated does an organism have to be to feel pain?
Yeah, that's a good question. That's a tricky question. So in other words,
it's a tricky question.
When you experiment with a worm or a fly, is it sensing pain?
What do you think? What do we know?
What we know is that it has some of the fundamental elements of what we call nociception, the ability to detect different types of stimuli.
I mean, we can see with our own eyes, though.
Yeah, and it has a defensive reflex. So in that sense, it is sensing things in the environment that can tell it whether it needs to escape from a certain situation. So yeah, I would say that, complex eukaryotes, as we say, not just vertebrates, but, even a fly has some sensation like that. The question is, do we regard that as pain like we do for a mammal? And there are different, views on that.
And how do you manage that, since you're studying pain in your own lab, right? With the creatures that you use, the experiments you do, the work.
So that's something that, so we use, for a lot of so one of the goals of our research actually has been to isolate the molecules that are involved in this, so that a lot of the research we can do that used to be done in animals can now be done in a petri dish. Right? So if I want to know how the capsaicin receptor works, I don't have to put capsaicin receptor on someone's arm or on a mouse's paw. I can now just ask what happens in a cell expressing this. And so that's a great thing for drug design and for basic research to understand these molecules, because it really gets us not only into a more reductionist system, but means that we don't have to, use animals for that. But there are some things where you do, and, you have to be very cognizant and mindful about what you're doing and reserve those experiments for the most important things and do them in a way that's as humane as possible.
Interesting questions here. Is it pain that drives evolution or fear of pain? How do you differentiate the psychological component from the physical reaction?
Yeah. Well, the physical reaction, the way that people often measure the physical reaction is through what we call a motor or a spinal reflex, right? So if you were to, for example, take your hand and put it on a hot plate, you would draw it quickly, or a mouse's paw would be on something hot. And that's really sort of more of a reflex response that's measuring the initial detection. You're not asking the person, how did that feel, et cetera. So that's how you can, and you can measure it electrically. You can record from the nerve fibers in a person or in a mouse or whatever, and ask if those nerves became activated when you stimulated, this, what we call receptive field, the periphery, where that your hand, the paw, whatever is coming in contact. So you can distinguish these things by measuring the early events, just by measuring electricity or other proxies of that, without having to ask anything about
There's some very interesting
sensation.
questions here about where the pain is and where we feel it and related. So one question is, what triggers phantom pain?
Yeah.
Another question related, why is it sometimes you feel pain in a different part of the body from where inflammation occurs? For example, pancreatic pain can be felt in the shoulder.
Right. Yeah. So things like what we call referred pain, like you have a heart attack, which is basically pain in the lining of your heart, the pericardium, and you feel it in your shoulder. That's called referred pain. And that's at least in part because there are some neuronal branches that have shared connections. And so, you may have pain in one part of your body, but that message is also being sent to a region in the spinal cord that's being innervated by multiple inputs. And so it's hard to disambiguate.
So
where
is
that
it
pain
confused?
Yeah, it's sort of a convergence of receptive fields, right? So it's a convergence of receptive fields from, say, this region and from the region around your heart. And so sometimes it's hard to disambiguate those things. And one place where that's particularly difficult is in visceral tissues. So like, sometimes when people have gut pain, they know they're having gut pain, they know it's kind of in their gut, but you don't know exactly where it is. And like when, young kids have appendicitis, sometimes it's pretty hard to diagnose because they haven't yet, they can't localize the pain.
Well,
very
let me
well.
ask the audience a question. Who's had a toothache? Okay. Who's had a toothache and you go to the dentist and they actually tell you that what you're feeling
Yeah.
is someplace else?
Right.
Right?
Yeah.
Referred pain, right?
Right.
Why
Referred
is that happening?
Yeah, because sometimes the
What's
circuits are shared and they converge at some point, and you can't. But the other reason is that you may have some inflammation going on that's sort of distributing the pain too. But a lot of it is just that you get a general idea for where it is, but there may be some shared inputs so that the messages converge at a certain point, and you can't, roughly where it is, but you can't exactly pinpoint it. And that depends upon the circuitry, but it also depends on the density of innervation.
Okay, this next question doesn't have the most votes, but I feel compelled to ask it. Why do men have such a lower tolerance for pain?
If we're wimps. Yeah, well, it depends on the pain, right? So as we talked about visceral pain, women have more of an experience with that.
Is there an evolutionary component to
Well,
that?
actually, yeah, I've talked about that a lot with my wife. So in visceral pain, what one could imagine, and especially since that's experienced mostly in women of reproductive age, particularly, is that, it's probably, or one could imagine that it's a mechanism to protect the fetus and to protect the act of reproduction, so that you have to be sensitive to and guard against things that you might eat or come in contact with that could be toxic. And so that's one particular idea, that it's really to guard reproductive health.
Interesting one here. How is acupuncture related to pain sensation?
Yeah, so there's a famous study that was done at UCSF many years ago by someone named Howard Fields to suggest that things like acupuncture are, so the experiment was, and I think it was acupuncture, is that the effectiveness of acupuncture and some of the treatments can be diminished if you take a drug called naloxone, which, as many of we know, Narcan, blocks the actions of endogenous opiates or morphine, for example. And so that led to the idea that some of these, say, non-traditional treatments, if you want to call them that, harness the power of your endogenous opiate systems, and that they may be, promoting the release of opiates that lead to a decrease in pain sensation.
Have you done research on that?
We haven't, no.
Do you want to?
No. I mean, I think it's fascinating, but one has to focus, right? And I think it's fascinating, and Howard and others have done a bunch of research on this, and so that raises the question as to can you harness these types of endogenous systems, and I think that brings to mind some other thing that maybe we touch on, which is placebo effect. So that's what this really was. I don't know if it was acupuncture, but it was about the placebo effect. And so there are endogenous systems in your body that, allow you to deal with pain. I mean, endogenous opiates are definitely one of those, when you're, there's that famous writings of Livingston when he was attacked by a lion. And he said, the lion jumped on him and shook him around like a rag doll. And he said, in his ultimate mercy, God provided me with this endogenous system. He's talking about opiates. So he said, I felt no pain until, later on, even when my shoulder was crushed. And then, of course, the pain came rushing in. So, at certain times, the opiate systems will kick in. And this is a great thing, but it's also a difficulty in drug development because the placebo effect is rather high for testing drugs. And this is a real confound and so there's at least two issues. Again, this is not my area of expertise, but something I've read a little bit about, which is, in drug trials, first of all, is there, you're asking people about pain. It's very subjective, right? So, how do you objectively measure? Are there ways to more objectively measure pain? And the other one is, how do you deal with the placebo effect, which could be quite high in drug trials?
I do want to ask you about that. Again, I'd ask the audience first. We have just a couple minutes and then we'll wrap up here. But how many of you have been asked by a doctor or somebody else when you're explaining about that you've got a painful experience on a scale of 1 to 10, where is your pain? Right? Do you like that question? No.
Yeah.
How do we objectively measure pain?
Not real well. And
but that's a very typical question, right?
Yeah, everybody, if you have a kidney stone, it's 10, right? If you have a toothache, maybe it's, it could be 7 or 8, I don't know. But so that's a big question in the field. And I think, people are thinking about ways to do that, looking for biomarkers, which is I think a long road to hoe, molecules that may be produced that are indicative of your level of pain. And the other one is imaging. To use imaging of brain or spinal cord or other areas to be able to see what regions of the nervous system are lit up without having to ask people in a declarative way, what's your pain like?
I want to cluster a few questions here because we're almost out of time, but they're fabulous. Is fibromyalgia a real disorder? Is one question. Another question is, does psychological or emotional pain show up with the same receptors as physical pain? But let me.
Okay, so psychological or emotional pain, and again, we're getting a little out of my territory as more sort of psychology, but I think that's mostly the domain of the central nervous system of the brain. And so, would it involve the receptors we're talking about? No, you can probably have that kind of pain without ever activating the receptors that we're talking about. That's a different sort of emotional pain. And so I don't have much to say about that in terms of what we've worked on. Fibromyalgia. Yeah, I think it's now recognized as a pain syndrome, and there are some drugs like gabapentin that help reduce that, which is sort of part of the evidence that, there is a physiologic thing going on. But this is, I think this points to a whole issue in the pain field, which is why we need more objective measures of pain, because a lot of people complain about, so the issue with chronic pain that's sometimes very confounding, and visceral pain is one example of this. So, you know, acute pain, you know what happened. You know, you whack yourself with a hammer, you touched a hot stove, you touched a piece of, you know, really cold ice, something cold on a, you know, -20 day in Chicago, you're in pain. Chronic pain can be different because let's take, you know, IBS, inflammatory bowel syndrome. The most common instigator of that is something like severe bout of food poisoning. Okay, so you know what happened. You know, you have these bad bacteria in there, all kinds of inflammatory stuff's happened. The bugs have made all kinds of nasty things. You know, you're in pain. Okay, that's fine. Then all that goes away. You know, the histology in the GI tract's looking good. You know, doctor looks up there with the scope, everything looks good. And you still say, but I'm still in discomfort and pain. I'm like never the same. And this can go on forever. And then, you know, people get, you know, a little despondent because doctor says it's nothing, you know, we don't see anything. And that's true for a lot of pain syndromes. So, you know, there's now an absence of a physical manifestation of the pain, and yet you're saying you're still in pain. And that's probably because there's a lot of things that have happened. You know, there's activation of nerve cells that have led to a continuous activation somewhere in the periphery or in the spinal cord. Same thing like, let's say, talk about, you know, phantom limb pain or in the brain, and you're left with this persistent pain syndrome, and yet the peripheral signs of tissue injury or destruction are now resolved. And so this is very frustrating to patients because, one, the doctor doesn't know where the initial thing might have happened, and then, you know, they don't know exactly what's going on. So having more objective measures for pain, although I think we're, you know, quite a ways from that, you know, if that could be achieved, would, I think, really not only help the diagnosis, it would help the patient, and it would help assessment of drugs.
We're about out of time. Last question here. I study pain, why not pleasure?
Well, nobody ever complains about too much pleasure.
Before we wrap up, you and I were talking earlier, not only about the importance of curiosity-driven science, but also the importance of the funding of science and where the field is and what the field needs. I'd like to give you a minute here as we conclude to share some of your, it's not often we get to share a stage with a Nobel and Kavli
exactly.
person. So what are your...
Well, thanks for asking that. So, you know, most of the work in my lab's funded by all of you, all of us, the taxpayers, through the National Institutes of Health. And it's really, you know, especially after World War II, the NIH sort of really became a powerful institution, and it's the main driver of biomedical research in this country. And it's really what's, and I'm not just saying because I'm funded by the NIH, but, you know, it's what put me through graduate school. It's what has trains people in my lab. Not entirely. We have also some money from private foundations, which is greatly appreciated, somewhat more flexible. But it's really the NIH that is responsible for the US being the preeminent place in the world to do biomedical research. And, you know, the great thing I think about the wisdom of the NIH is, yes, there is a big focus, of course, on health and disease and some targeted funding for treating cancer or, you know, HIV, whatever. But there's also sort of a layer of the NIH that just recognizes the fact that you have to fund curiosity-driven research because it's the basis for everything we know. And there's no predicting what's going to happen. I mean, you know, everybody now talks about things like CRISPR is being used for this, you know, amazing new technology to edit genes and cure things like sickle cell, whatever. You know, that wasn't discovered because someone went out and said, gee, I want to discover a new process for editing genes for gene therapy. It was because some guys in the lab and gals, you know, were playing around with bacteria and noticed that there were these weird things happening in their system to protect them from invading viruses. And they understood all the, you know, they started identifying the pathways that are involved in that. And that's what emerged into CRISPR technology. And, you know, all of the work that people like me and others as graduate students spent years understanding how enzymes work, like proteases and enzymes involved in DNA and RNA replication. That's why we have drugs to treat people with HIV. Because all of that early work and just, you know, curiosity, like how does this molecule take a substrate, you know, and turn it into something else? And what are the, you know, all the nerdly things, right? What are the
Right.
atomic bonds that allow this to happen? And, that's what drives technology, and that's what drives the development of drugs and industrial science, is that basic understanding. If you don't have that, you may get lucky on occasion or find a natural product, but it's not kind of the process that really leads to long-term sustained progress.
David, thank you for your work, for your brilliance, for your contributions, for your insight. And it's been an amazing conversation. We could go all night. I think we may have a chance for a few more questions or whatever later. But Eric, let me turn it back over to you with thanks
Thank
to
you.
David Julius.
So, David, thanks again for your work, but also for the clarity with which you conveyed that to us. It was remarkable. And I bet everybody understood everything you said. Indeed, it's a very complex topic. So thank you for that. Frank, thank you for as always, a great discussion. You seem to enjoy this one a lot, maybe most, I don't know. But
No game.
yeah, but we always appreciate it when you come around and do this kind of thing. It's just amazing when you think about the stuff you're doing, David, and how the evolution has pulled together all these tools. And basically, it's very clear pain is important. So before we finish, I just wanted to also Kathy Bailey, are you here somewhere? Kathy, there's Kathy. I just wanted to thank her. She's the director of the Kavli Prize, so she has a lot to do with it. She's representing Kavli today, so I really appreciate you being here. Thank you. I also thank the Kavli Foundation and Cynthia. And Petra, yes, thank you, Petra. Sorry. And the Norwegian Academy of Science and Letters, and as well as the Norwegian Embassy here in the US. And I think, I'm going to walk away from this with a lesson that when you go home tonight and you slam your finger in the cabinet, you shouldn't be cursing. You should be grateful that you're feeling that pain. It's what we survive on. With that, I want to thank the audience for being here, the great questions that you gave to Frank and to David. And we'll see you again in the fall. We'll jointly host another one of these Kavli lectures. Thank you.